Good morning. I'd like to begin by introducing Dr. Anna Ritchie. Anna is my consumer buddy. She has MS. She was a general practitioner until she had to stop her clinical work as her MS developed. She's mum to Eliza and Ellie, two beautiful little redheads, and wife to Ben, and her mum is here today as well. And she's a knitter. She's a strong advocate for women's health and has been involved in a program called Knit Your Bits. And there's a photo here of Anna with some of the bits Anna has knit.
Did it work? Put it to the back. Can you make the thing work?
Yeah, here.
Okay, thank you. This is Nick. Nick is a computational geneticist. He grew up in Tassie and he did a science degree at UTAS and then developed an interest in human genetics at this time. He went on to do a PhD in inherited cancers. Can you not hear me? Microphone. Okay, sorry, I can hear that it's working. So Nick did a science degree at UTAS and then went on to do a PhD at Menzies and in inherited cancers. Then he went to America and worked for 5 years there in different genetics projects, and then came back to Tassie, was recruited back to Menzies in his current project. And when he came back from America, he brought with him his partner Ignacio and their cat Zondli.
So why are we working together? So for me, it's about creating a partnership with a consumer, someone who has MS, and involving them in my research. Having this back and forth discussion about what my research is and what it means to a person that has MS. And I get the privilege of learning from someone with lived experience, someone that has MS and is living with this disease.
For me, I like science. But this allows me to not just be a passenger to that science, but to be a driver of it as well. The back and forth is really important to me as well. I get to hear what's going on for Nick, go away and talk about it myself and think about it and come back and clarify things I don't understand. And then I get to ask the why. Why does this matter for me? Why does this matter for the community? Why does it matter for my children and the next generation?
I'm usually pretty good at this. All right, so what have we done? We meet regularly for coffee or tea for Anna, because she's not addicted to coffee like me. We discuss Anna's journey with MS. Anna would describe herself as fairly new to MS, which is a unique opportunity for me as a researcher because I've joined her on this journey fairly early on. She's still learning how MS feels for her. As new symptoms arise, is it a relapse coming, or is it just disease as it is now? Anna's reviewed research documents like participant information sheets and provided input to them. She's also been instrumental in generating lay summaries for grant applications and contributed one to my fellowship that was successfully funded by MS Australia, thank you, at the end of last year. For me, I get to have a person that I can think about the context of the research that I'm doing. I'm not a doctor, I'm not a clinician, I don't see patients, I don't have a group of people with varying symptoms at different time points of their disease to think about and understand MS. So Anna has given me that opportunity to understand what MS looks like. Not that you... And we also think that we hope that our partnership has shown others in the flagship and within the Institute how a research buddy situation can work and the kinds of things that can come out of it.
So what is the project?
What is the project? So the project I work on is... if I just throw it, the MS family study. Rach, this is not, okay. We look for families with 3 or more close relatives that have MS. And we study these families to find genetic changes that are shared by affected family members and not in their unaffected family members.
So, can I stop you there? Something that I want to know at this point is why? Why this project?
So why is a very important question, and the answer is we don't know why. We don't know why these families have this unusual level of multiple sclerosis in them. 1 in 4 to 1 in 6 people with MS have a family history, but families with 3 or more people with MS are rare. We believe that studying these families can help us understand why they are at a greater risk for MS at a genetic level, which can give us clues as to how MS starts and hopefully a clue as to how we can end MS.
Okay, great. So, what are the steps? It's working. So, first we have to identify and recruit the families. So, it's the neurologists who identify the families, and they ask if they want to be involved and then hand them on to you. And then you take the family history. So, my grandpa had MS.
So let's take your family history.
Great.
So here's Anna, a dot on the screen. I'm going to draw a pedigree here. In this pedigree or family tree, circles are female, squares are male. If there's someone who's darkly coloured in, they have the disease in the family tree, so in this case MS. So Anna, do you have any siblings?
Yes, I have a twin sister and a younger brother. Maybe. Eventually.
Right. Now, I've met your mum, so I assume you have parents.
Yes.
Now, does anyone in your parent know?
They
So
don't
none of them
have
have
MS.
MS, it's just you. So you said that your grandfather had MS. Was that
Yes.
on your mum's side or your dad's
Yes,
side?
that was my mum's dad.
Okay. And is your mum an only child or?
She has 3 sisters.
3 sisters, and do any of them have MS?
No MS. No.
No MS, okay. So your family is not the right family for the study that we have, because there's not quite enough MS in your family.
Okay, so what would it look like if we could be recruited to
Okay.
this study?
So if your family had 2 more living people in the family with MS, for example, your mum and one of her sisters, or your sister, we would recruit everyone that has an arrow pointing at them into this study.
But not my brother or sister then?
No, so you look like you're under 50?
Yes, I am.
Yes. So your twin sister is, of course, the same age as you, and you said your brother is younger. So we
Yes.
don't recruit any unaffected family members that are under the age of 50 unless they're the parent of someone with MS.
Okay, great. So what are the next steps? Next we have to collect samples. Are you happy to do
Yep.
that? So these are our new study participants. We need to collect samples from them. So we can collect saliva or collect blood. You prefer to collect blood, you get a better quality DNA sample from that. But it's important to have the alternative of saliva. People who really don't like having blood taken, I'm looking at Jane who's just said that she... So they can have a saliva sample collected, but also people overseas can have a saliva sample collected because you can post saliva but you can't post blood. Then you have to get the DNA. So you put the sample in the machine and it spits out the code. Now, there's another reason that you like to collect blood. That is because people can also join another study happening at Menzies called MS STEM. So the blood sample, the blood cells get collected from the blood sample. They then get them dividing in a dish. I know that Kayleen's watching me, I hope I get this right. They get them dividing in a dish. They then introduce a virus which has been programmed to change those cells. And as they're dividing, and with a little bit of magic, and as they're dividing, they turn back into stem cells. They then add a bit more magic, but no virus this time, and they can turn those stem cells into different brain cells, and the brain cells can be studied, and those brain cells have the same DNA as the person who donated the sample. So what are the next steps? We've got the DNA code, maybe. Now, if you ask the computer to print out the code, this is what it would look like, if it appears. Now, obviously that's very complicated and nobody wants to look at that themselves, and that's why Nick uses a computer.
So when we've sequenced someone's genome or read their genetic code, on average we find 3 million changes to their genome between each of us. So here's our example family where we've got 3 people that have MS and an unaffected person in the white there. And here, if we just represent with these coloured dots all their genetic changes that we've identified. So there's a lot, but we have some public databases of genetic variation from thousands and thousands of people that let us know the frequency of genetic changes in the population. So if we see a change in one of these databases that's fairly common, we can assume that that change is not the underlying reason why that family is developing MS, so we can exclude all the changes in that public data set from our analysis. Now in this family we've got 1 unaffected family member, so any genetic changes they have that they share with their relatives, we can exclude those genetic changes from our analysis, leading us to just 2 changes in this family, or the interesting bits to focus on. So then we go in deep on these 2 changes to understand what genes they are affecting, and then trying to understand how that change can impact the cells that are important in the development of multiple sclerosis. So what have we achieved so far? So we've recruited and sequenced 8 families into our study, and these range in size from 3 family members with MS through to 6 family members with MS, as well as 3 to 4 unaffected family members. We've sequenced over 40 genomes across these 8 families and identified 8 changes, the interesting bits that we're focusing on now in the laboratory to understand what they do.
So is that 8 changes in 8 families?
No, so that's 8 changes in 2 families. So we're still analysing the remaining 6 and taking our best clues forward into the laboratory and further analysis. We see one family has 2 changes and the other family has 6 changes, and each of those is what we call a hypothesis as to why, or a best guess as to why MS is developing in that family. So we're trying to figure out if that is true or not. So what will the future bring? So each family gives us clues that we can try and understand why MS develops in that family. But those families are rare, and we want to extend these findings to the broader population of people with MS. So we might see the same or similar changes occurring in the broader population of people with MS, and that can give us clues as to where we should focus our targeted drug development. So
for all these people here, when might they expect to hear the outcome of this? When might we expect to see it in the clinics even?
So research is slow. It takes time, and this is discovery. So we're right at the start of the pipeline. But things can move quite quickly once we have something to latch onto. And I think we're almost at the point, Kayleen's nodding, that we've got something to latch onto. So I think in the next 5 to 10 years, we're going to have something to take into the clinic in a really targeted, specific way. Okay. It might only benefit a group of people with MS, a specific group whose MS is developing for a specific reason, but even that is a massive step forward. So these changes, once we've found them, it's not direct from the bench to the bedside or to the lab, to the clinic. We need to understand how they are causing MS, and this is the basic biology. What cells are involved and what are these changes doing to them? And part of that is our MS STEM study, which is separate to MS family, but most of our families are involved in MS STEM. And that involves a large group of people that drive this research forward and our understanding of these changes in these cells. So it takes a team. Can we go back a slide? Thank you. So consumer involvement involved in this project has been across the entire project, and here is one consumer that's been heavily involved in this project as my research buddy. Anna's and others of the Consumer and Community Reference Committee have reviewed participant information sheets as well as plain language summaries in helping us as scientists communicate the research. Often we know what we want to say and we know how we understand it, but we don't know how to say it in a way that you all understand it. So it's people like Anna that help us translate our research out into the world and spread the message to everyone. So this project has not been would not be possible without the families who have been involved, so there's MS families, many researchers across the flagship, as well as our Consumer and Community Reference Group and Committee, and Anna, as well as our project funding from the Irene Phelps Charitable Trust and the MRFF, as well as NHMRC, the Royal Hobart Hospital and MS Australia, who each fund different aspects of this large body of work. Thank you.